Thank you very much for joining us in person or online tonight. This is our semi-virtual community lecture series as we all are navigating how to come back into the real world after the last 20 months or so. We are really pleased tonight to have actually two terrific lecturers who I think are going to give a great presentation tonight. Before we talk about that, I just want to finish my welcomes. Of course, thank Kyle Good, who's at the back of the room and obviously made all of this happen. Brian Cummings, of whom all electronics are terribly frightened. He can just look at something and make it work. If you have problems at home, let us know. We send him out for hire. He also organizes our community outreach program, which is great. Judy Beck is our director for communication. She couldn't be here tonight. And with that, I just want to highlight, I'm on a very short program tonight because I really think there might be some interesting questions for our presentation, not that there aren't always from our interactive audience. Just want to highlight how can you help, how can you be involved with the center, sign up for our newsletter, follow us on Facebook, follow us on Twitter, go to our homepage. I'm going to show you that in a second because our October newsletter is up. And of course, research, drives certainly our economy in California. We're very grateful to have you all interested in this series. But of course, we need your support as well. So think of us as you're considering making philanthropic gifts. This is our current website. I just really want to highlight that you can find here our recent recorded lectures if you want to send that on to anyone who might be interested. And we have quarterly newsletters now. Our most recent one, which is for October, is up and live on this website. So I invite you to go there and take a look at what's happening in the center. I also want to call out a couple of save the dates, which are important. January 11th at 7 o'clock, our regular timing. It's a Tuesday coming back from the holidays. We are glad to welcome Lee Turner, who is a new recruit as a faculty member. He's at the back of the room. Give us a wave, Lee. You'll show up on the Zoom, in fact. We're really excited to bring Lee here from University of Minnesota because he studies ethics and stem cells combined. He has lectured for our scientific audience, our faculty audience at UCI several times, which is why we snatched him away from the middle. And he's come here as professor of public health and to direct a new center for bioethics. He's going to be speaking on the American stem cell. He just had a big publication accepted in Cell Stem Cell, which is, I think, coming off embargo on Thursday. And one of the things that he works on is how US businesses are marketing stem cells and unlicensed and unproven therapeutics. So this is a really great one to catch, and I invite you all to participate. Again, you can register on our website and at the same site. I also want to call out a save the date to the big evening lecture that we do every year. Unfortunately, last year, of course, it was completely virtual. This year, we're angling for a hybrid mode, so we will be hosting an events permitting, at the Irvine Barclay Theater. That's going to be Thursday, January 27th at 7 o'clock in the evening. These are really great events. Anthony Atala is our keynote lecturer and our evening public lecturer. He has an incredibly stellar career in stem cells and bioengineering. And so I invite you to have a look at this on the website and please consider registering and attending in person because we hope that's going to be a great one. With that, I just want to introduce briefly Tim Downing and Mike Zaragoza. Who's going first?
We both are.
Excellent. That's Mike's theme. So Tim Downing and Mike Zaragoza. Tim is, I think, probably in the tenure pile right now. Yep. Wish him luck, everyone. He came here with a bachelor's from Northwestern University. Interesting tidbit about that. He actually was an undergraduate researcher in a laboratory that Brian and I have collaborated with for almost the last 17 or 18 years, which is just shows you how small the circle in science can be. He went on to get his PhD from UC Berkeley and did a postdoctoral fellowship at Harvard. He came to us in 2016, and he has a number of distinctions to his name, including Forbes 30 Under 30, and he's been named as an NIH New Innovator Award. And so we're really pleased to have him here at UCI, and he's been incredibly successful. Mike Zaragoza has awards that are actually too numerous to enumerate here, particularly for teaching. This is something that is, he really excels at in terms of the medical curriculum here. And he's a trained geneticist and MD PhD. So he's been a wonderful addition to UCI, where he arrived, I can't see your line. When did you come? 2000, there you go, right after receiving apparently his MD and PhD from Case Western. He works on patient-derived induced pluripotent stem cells and studies the genetics of inherited cardiovascular disease and how we can work towards therapeutics in that regard. So a couple of quick words on format with those brief introductions. Mike and Tim are going to go ahead and give their presentations. Clearly going to be a very interactive one since they're organized and going simultaneously. I'm going to ask everyone to hold your questions until the end. It'll be possible to submit questions from the audience who's online through the Q&A box. We'll be monitoring that. I'll be looking at it on my screen. People will potentially be shouting out questions to me to make sure that we're getting those answered as well. And of course, everyone in the audience here tonight, we're really glad to have you participate. Thank you for your patience.
All right. Well, thank you, everyone. This is actually one of the first lectures I've given live since pre-pandemic. So thank you. And Tim and I, we've known each other for many years and actually collaborators. So this is going to be fun for us. And, you know, Tim, first of all, I have to say that we've already stopped the hands of time, because this is a picture several times, and one more time because this is a picture of me about 12 years ago. Okay. He finds that funny. That was not supposed to be funny, Tim. And how old were you in this, or how long ago is this picture for you, Tim?
This is assistant professor year 1, 6 years ago.
6 years when you were not too stressed and energetic coming into the system here. Anyway, so we're going to go over stem cells and aging. And why would we really want to talk about this topic? Certainly it's very important, okay, because this topic affects you know, our life, our lifespan. And just going into some of the data from the CDC, this is a graph that you might have seen. It shows on the bottom the year, as well as the age on the other axis. And then it plots out the US life expectancy at birth. So this is the average lifespan that you would expect at birth. And just for comparison, you can see it in 1900, that age was 47 years of age. And so just with 100 years, at the year 200, that jumped up all the way to 77 years. So that was a dramatic increase in life expectancy. And certainly, as you know, that was due to public health and also a medicine for infectious disease. But you can also notice here on the more current graph that shows from year 200 to 2020, the last 20 years, it's going from 77 to about 79. So we haven't made that much gains over the last 20 years. And the reason behind this is the shift from infectious disease to chronic diseases. So we haven't made much of a gain. And of course, if you update the slide all the way to year 2020, you actually see the dip, okay? And this made a lot of news because, of course, what we're all dealing with, okay? But the point is that over the last 20 years, we made some progress, but still, lifespan is still limited. So what we're going to do, we're going to talk about, first of all, what is aging? Dr. Downing is going to talk a lot about stem cell epigenetics. How do we measure aging? That is, how do we study aging? And then finally, we're going to go into some examples of possibilities of how to treat aging. First of all, to start off, I just want to ask everyone, well, what is aging, first of all? So just so we're on the same page, what do you think about when you think about aging? A decline, definitely. What else do you think about? What happens when you age? Who ages? Any thoughts? Oh, who's aging here? I'm raising my hand, okay. Tim, what are your thoughts about aging?
Yeah, I think it's a constant decline of the human body.
Exactly, right, thank you. He saw the slides. And, so aging is a process, and I'm going to go through some of the medical definitions, okay? But but we do have a lot of thoughts about this. But in general, the definition of aging is a functional decline of the human body, okay? And what is the human body? Human body is made out of cells, okay? This is the basic building block. It's like the bricks of a building. Okay, and these are the functional units that make up the human body. And then from these cells, they're grouped together with a shared function for tissues. And then there are more complex structures called organs that also do specialized structures. So this slide shows how, during development, you start as one cell all the way into a very complex individual, the human body, which is actually made up of over 50 trillion cells, and then over 200 different cell types. So these cells are very specialized, as listed here, and they're mature. Most of them do very specific functions. So these are heart cells, skin cells, and then as we age, unfortunately, there's a functional decline in the cellular process, the tissues, as well as the organs. And this is a natural process that we would term aging. But I also want to bring up the question, is aging a disease? No. Who here says aging is not a disease? Just a show of hands. And that's pretty much the majority, okay? And why is that? What is that? Why would you say that, okay? Because this is actually a very controversial topic when you talk about this, is it a disease or not? Why is it not a disease? It happens to everyone, absolutely. Any other thoughts? It's a natural process. So at least medically, we define disease as an abnormal process, okay? So you'll see a lot in medical literature as aging as defined as a natural process that, whether you like it or not, happens to everyone, okay? So when I think of aging, I think of what's one of the first signs of aging? Losing your hair. And what else? I think of vision, okay? This is where my vision, the lenses get thicker, so you can't focus on objects which are close. I refuse to wear bifocals, but still, that's one of the signs of aging, the functional decline. So these are natural changes that we can go through every system, where some are actually, early signs of aging, others, seem to more stable. Now, the second point, whether or not aging is a disease, so most of the time you'll see it's treated not as a disease, okay? But there's also the flip side is that, well, if you treat it as not a disease, then should we treat it, okay? I mean, should we investigate it, okay? And I think the reasons to investigate aging is not that it's a disease, but it's a known factor for increased risk of chronic diseases. So if you don't think of it as a disease, it's certainly associated with chronic diseases. So in this slide, this is a cohort which shows age in years in the bottom, and then the incident rate of different chronic diseases in a population. And it shows just the different types of chronic diseases associated with aging. And that's from the top: cancer, heart failure, COPD, which is like emphysema, chronic bronchitis, myocardial infarction or heart attack, stroke, dementia, and diabetes. So these are age-related diseases that, as the population is aging, the incident increases, actually exponentially. This is on a log scale. So you can also notice as we get aged, as you would expect, that the mortality rate is also increasing at the same rate. So the point of this is aging is one of the main factors of chronic diseases. Okay, so if you don't think that if you don't think aging is a disease, it certainly contributes to as a factor for chronic diseases. And then other things. So we like to study aging, not just because, certainly we want to extend health span, but also understand how aging contributes to different chronic diseases, especially other ones listed on this slide like osteoporosis, stroke, arthritis, sarcopenia, which is muscle wasting, changes in your vision, vascular disease. So next thing I want to talk about is what causes aging. Now, this is a very broad and deep topic because it's been subject to a lot of different research. So some of the hallmarks of aging are listed on this slide, and the ones that we're going to focus on tonight are stem cell exhaustion, which essentially is as you get older, your stem cells burn out. Okay? And the other thing that Dr. Downing will go into more detail is something called epigenetic alterations. That is the changes of your DNA that contribute to the aging process. So back to the slide with the tissues and organs, and how does this change with age? Certainly, we have trillions of cells that make up tissues and organs, and most of these are mature, specialized cells. But how do these tissues and organs replenish themselves? So this is a slide that shows the different types of what we call adult or tissue stem cells that are less mature, and they have a certain ability to renew the cells within that tissue or organ. And so what happens during aging? And you've probably heard this attending some of these lectures. What is the definition of stem cells? And this shows for these tissue-specific stem cells or adult stem cells, there are these quiescent dormant stem cells. And then a stem cell is defined by two characteristics. One, its ability to self-renew, that is, divide and then replace itself. And then number two, once it's activated, its ability to differentiate. That is, you have this very immature cell and becomes more specialized. Then these can give rise to other progenitor cells and then eventually differentiate and become highly specialized and mature. So for example, for your blood cells, you have hematopoietic stem cells in your bone marrow, and then the blood cells last roughly, I think, about 120 days. So as those get old, the blood, it has to be renewed. So your stem cells would be, which are initially dormant, would be activated, self-renew, and then differentiate into the different lineages, the mature cells in your blood. Not only your red blood cells, but also your white blood cells. So in that sense, this system allows you to replenish those cells that have a limited capacity, to age. Now, this is the concept of tissue homeostasis. So as we get older, this allows us to replace the cells that die. It also allows us to be resilient. That is, if there's stress or injury or infection, it allows the body to recover by having this sort of backup cells that could not only self-renew, but also replace those cells which are injured or dying. So there's this balance or homeostasis. Now, what happens with aging? Now, in aging, these stem cells can undergo many different changes. That is, they can stop dividing, they can not able to self-renew or replace themselves. They can be failed to be activated. There's also processes called apoptosis, which is programmed cell death, and then also a process where they just would stop dividing, or senescence. Now this picture, shows that process as we age, but also remember this is not in isolation. Not just the stem cell itself is important, but also the local environment of the tissue and organ that's also going to be able to allow this process to continue. So as we age, we're actually losing this backup system, the homeostasis, which results in the functional decline. Excuse me. Okay, so back to human cell. well, why does this occur? And so I'm going to bring you back to human cell and show you one of the compartments of the cell, which is called the nucleus. And going into some of the biology, a very simplified slide, as you remember, the cell contains a nucleus which contains the blueprints. That is the DNA, the genome. The genome is comprised of the chemical called DNA, which is packaged into different segments called chromosomes. So you could think of the DNA as the blueprint, chromosomes as the books, and this gives us the instructions, that is the genes, which are over 20,000 different genes that are then translated into proteins to carry out the functions of the cell. And the cell is very mature, so you can imagine different genes are activated to allow the specialized functions for each of those cells. Now, this is a very simplified diagram known as the central dogma. And then here I'm going to kind of hand this over to Tim, because he's going to go into a more complicated process called epigenetics.
Awesome. Thank you, Mike. I think everybody can hear me okay. So like Mike was mentioning, this sort of picture of the central dogma, my lab is very interested in sort of this central question, which is how do a single genomes, knowing all the cells in your body have the same genetic material, the same ATCG sequences, how does that single genome beautifully give rise to all of the cellular heterogeneity that exists within our bodies? And the field is really starting to understand that a large way in which this is accomplished is through what's called epigenetic regulation. And so epigenetics refers to modifications that happen, as the name suggests, on top of, or epi, on top of the genome, and include things like histone modifications that are within nucleosomes that wrap around DNA. These modifications play a big role in the accessibility of certain genes in the genome and whether they can be read out and expressed, ultimately giving rise to that protein function that allows those cells to specialize. Also, DNA methylation, which is one that we'll focus particularly on today, and I'll explain what DNA methylation is. But I just want to highlight back to this point that Mike made about stem cell replication, and how replication itself actually seems to play a big role in the way stem cells are moving through different states or during the differentiation process. And what's always remarkable to me is that these cells need to replicate their DNA sequences to copy and make two cells, but they also need to replicate their epigenetic landscape, all the DNA modifications that are there, the histones, the modifications to those histones, in order to remember which genes to keep on and which genes to keep off to sort of maintain fates. And this process of replication can actually help, again, these stem cells move through different states. And so here's sort of a classical example of what's referred to as asymmetric cell division, where during division of a stem cell, it can give rise to two different types of cells during differentiation, or obviously self-renew. But also there's population level heterogeneity within stem cells that are cell cycle dependent. So if you take a population of stem cells and you expose a single cue to those stem cells, they all don't do the same. They don't behave in sort of a deterministic fashion where all of these cells commit to the same fate. They actually have different programs of activation based on some type of intrinsic heterogeneity that exists within the stem cell population. At the same time, replication is known to drive the process of aging, okay? The sort of replicative aging, as it's called. One of the sort of more familiar examples that you might be familiar with is with telomere shortening. So the chromosome ends get shorter as cells replicate, okay? And this can contribute to genomic instability and ultimately facilitate this aging process. My lab is very interested in how epigenome replication actually contributes to these seemingly disparate processes of life, both allowing and enabling stem cells to differentiate and give rise to all of that beautiful cellular heterogeneity that exists within your body, but also how it might confer susceptibility to drift, or basically the accumulation of errors in the epigenetic landscape as we age. Okay, and so we theorize that temporal delays in epigenome replication serve really two functions. They give rise to regulatory variation, and so noise effectively. And so like I mentioned, that intrinsic variation. Yes? No problem.
I'm just wondering if you could explain for a moment about what you mean by cellular heterogeneity.
Yeah. So what I mean by cellular heterogeneity, right, is that if you have a population of cells, okay, all the cells don't have the same molecules within them, okay, and the same modifications to those molecules within them. There's variation across all of the cell population, and so that actually is what I'm referring to as heterogeneity. I hope that.
So if you have skin
Mm-hmm.
or cardiac tissue, for example, the different kinds of cells that make up that organ or tissue as a whole, that's one part of heterogeneity.
That is one part of heterogeneity, yep. And the other part of heterogeneity is that even in what we think of as a homogeneous population of stem cells, like embryonic stem cells, there can be slight variations between those cells as well that we don't fully understand, but that our lab has been exploring that can actually play a big role in the function of how stem cells actually differentiate. Great, thanks for the question. So looking back at this sort of picture, you know, and thinking about the duality of replication and how it's serving these two sort of purposes, we're going to talk today about DNA methylation, one of these critical modifications in the epigenetic landscape that actually is responsible for things like gene expression and gene regulation, but we'll see has a big, is largely implicated in the aging process. So when I say DNA methylation, I'm essentially referring to this chemical modification that happens on the cytosine molecule of DNA, so A, T, C's, and G's, this is this modification, but the more important thing to take away is that actually there's 56 million sites in the genome that can have some methylation mark, okay? So it's a location in the genome where it can have some DNA methylation, and 45 million of them will have some level of methylation mark present in your genomes. Okay, and so just to give you a sense of when I say to copy this information, these cells have to copy 45 million sites where methylation exists. So there's quite a bit of work that has to be accomplished for these cells to inherit. So thinking about DNA methylation in the context of aging, I think what's sort of one of the more notable things that DNA methylation allows us to do is actually to measure and get a sense of measuring biological age. So here's a plot of sort of a schematic of showing chronological age on the x-axis, but on the y-axis, we're showing an estimation of population level averaged age in the blue line here with chronological age, and you get some age estimation. And some people who age faster actually fall above this line, okay? And people who age more slowly fall below this line. Okay, and this can be, again, predicted based on, say, blood signatures in DNA methylation taken from a biopsy. Some of you may be familiar with companies that actually will give you your epigenetic clock age based on DNA methylation from your blood or other samples. So in my lab, we're very interested in actually studying and using human embryonic stem cells as a model to understand this process of replication and how epigenome replication might contribute to these two different processes of cell fate and also to aging. How we measure DNA methylation, we schematize it by just showing open and closed circles saying, for a given location, there's methylation present or absent in a given location. And so this, again, is sort of giving rise to some of that heterogeneity that I mentioned across stem cell populations from Eileen's question. And so in terms of, you know, replication and maintaining epigenetic modifications across replication, DNA methylation happens most frequently in a C followed by G context, and it has this what we call palindrome-like structure, because we know that C's and G's base pair with each other, and so there is this position where the cytosine exists on both sides of the DNA strand. One strand, both strands can have a methylation mark. And as DNA replication happens, okay, again, when I mentioned that inheritance, you have this substrate where the parental strand has methylation information that the daughter strand does not, and that information can be transferred through an enzyme that recognizes that substrate, DNA methyltransferase 1, okay? And so this DNMT1 enzyme will find that substrate and add methylation to the other side, okay? And in this way, across replication, you actually have a closed loop system where DNMT1 is bringing what is a hemimethylated strand of DNA after replication back into a fully methylated state, okay? And so this ends up being really important in thinking about replication and how it can propagate and how methylation modifications can propagate across replication. So again, cells can remember which genes to keep on and which genes to keep off. So the timing of this process was not very well understood, and so during my postdoc, we actually started some work to develop a method called Repli bisulfite sequencing. Long story short, it's a method that allows us to label newly replicated strands of DNA and capture and measure DNA methylation levels in those newly replicated strands over time. Okay, so we can see how methylation is reestablished in replicated strands of DNA over time. And with that technique, we were able to see that actually there are massive delays. The purple line is showing our newly replicated DNA, and the gray line up top is actually showing matured, fully matured DNA. And so there's about a, this actually shows a little bit better, about a 4-hour to 16-hour delay in the time that it takes for methylation to hit its steady state after it's being replicated. And so this blue plot on the side actually shows that what we actually hope to do with this information is actually to understand, well, there are some locations in the genome that are remethylated very quickly and come up very fast. Others may, come up more slowly. And we're wondering, can they have some implication or some impact on the regulation of genes, but also on the susceptibility of the epigenome to accumulate errors throughout many cycles of replication? So in the context of regulating gene expression, one thing that we wanted to look at is actually regions of the genome that are slowly remethylated versus regions that are more quickly remethylated, and ask, is there any relationship between the variation in gene expression that we observe across stem cell populations for these two locations? And the interesting thing is that when we actually do some computational sort of analyses, we can see that genes that have very large single cell variation or variability, shown in the red, actually have slower or the slowest remethylation kinetic rate, shown on the y-axis. If you look at cells with low cell-to-cell variation, they actually indeed also have higher or faster remethylation rates. And so the longer that these cells exist in this post-replication delay for a given gene around that locus, you actually see more variation in the actual expression of the genes near where those slow kinetics are, suggesting that there is indeed some connection between these post-replication kinetics and that functional readout. And so back when Mike was talking about that central dogma, what is starting to emerge in the field is that molecular variation or heterogeneity is very central to all levels of the central dogma, okay? You can see this at the protein level, where there's protein level variations between two pluripotency markers, also at the RNA transcript level, and even at the level of the epigenome in the sort of regulatory components of the genomic landscape. And so we think replication actually serves as this source of molecular variation across stem cell populations. Why is this useful? Well, stem cells really need to make fate decisions, not just in deterministic fashions, right, where one cue does one thing to all of the cells. They actually can achieve things like probabilistic differentiation strategies, where a single cue can actually help a stem cell to differentiate into maybe 3 lineages, where 30% of the time it'll differentiate towards fate A, 60% of the time towards another fate B, and maybe another 10% of the time self-renew. And this allows stem cells to give rise to complex tissue compositions that can then lead to self-assembly and remodeling that can lead to complex organ formation. And so this is one reason why we might have this intrinsic heterogeneity or variation across stem cell populations. So I mentioned this first part, what we think of as this source of regulatory variation and replication. The second part is it conferring susceptibility to drift in the epigenetic landscape throughout the lifespan. And so here's a data picture from a paper showing actually methylation of chromosome 8 across the whole length of chromosome 8 on the y-axis, again, that methylation level in a newborn individual shown in the purple versus a centenarian, someone who lives to be over 100 years of age, 103 in this case. And what you can see is that there's a global loss of methylation between these two individuals. And so this generally happens across all of our methylomes. If we were to take blood from everybody now versus 6 months from now, you would actually observe some losses in methylation as you are sort of aging. And so back from this model that I showed you of this closed loop system where DNA methyltransferase 1 brings hemimethylated DNA back into a fully methylated state, you can imagine a scenario where if DNMT1 doesn't have enough time to find that hemimethylated substrate before the next round of DNA replication, actually you have something called passive demethylation, where that nascent strand, what was the nascent strand, is now the parental strand in the next round of replication, and there's no methylation on that strand to inherit to the daughter strand, and so the methylation marks are fully lost. And so the idea is that as you're copying, you have this possibility for the accumulation of errors in regions that are very slowly remethylated across DNA replication. And so just to kind of bring it home, in our data, what we're seeing is that regions with increasing delays in post-replication methylation, so the largest delays here, actually are the locations that we also find have the greatest differences between young and old individuals in these blood cells. And so we can see some signature in DNA methylation that slow remethylation rates indeed leave CPGs more susceptible to losses throughout the lifespan. And so from a schematic perspective, we believe epigenome replication is sort of a two-sided coin, okay? On one side, it gives rise to regulatory variation, right? That gives rise to those variations in gene expression, allowing stem cells to do important tasks like probabilistic stem cell fate decision-making. But it also leaves our genomes susceptible to drift, the accumulation of errors throughout the lifespan across many sort of cycles of replication. An interesting study also observed that, epigenetic drift, how much of it is a driver of the aging process. A fascinating plot actually shows that different species will have different rates of epigenetic drift. And what you'll see is there's a linear correlation between the maximum longevity, or the number of years that species lives, and the rate at which their epigenomes drift. And so in the case of mice, they live the shortest, they have the fastest rate of change in methylation based on time. Monkeys are second, and then humans live the longest, and we have the slowest. And there's a pretty strong R-squared correlation between, again, this rate of remethylation and how long that species actually lives. So what might drive this? So we actually have some interest in some work looking at the impact of unmethylated DNA on our immune system. So you may be familiar with this term called inflammaging. Essentially, as we age, our immune system experiences this chronic low-level grade sort of inflammatory state that gets more progressively worse as we age. We also have immune cell receptors that recognize unmethylated DNA, unmethylated CpG DNA. We tend to think of those receptors as being for recognizing bacteria or viral genomes because they don't have CpG methylation in those species. But as we age, our DNA actually becomes progressively more hypomethylated, as I've shown, and we think that this actually makes the immune system's response to our self molecules, our DNA molecules, outdated as we age. It no longer sees it as self, and it starts to look at it as foreign and triggers this sort of inflammatory response. So this is something we're actively interested in, how we might connect epigenetic drift to the actual aging process. And then I'll just finish by saying, some theories on aging argue that aging emerged early in evolution as a means of controlling population size. And this is why we're really interested in this work, because you can imagine that if you have some population living in a local environment and resources can renew, but they're being consumed by the inhabitants of that environment, and without an aging mechanism, you consume all your resources, your population would crash out, and then you wouldn't really take the next evolutionary step. But with an aging mechanism, from a population level sense, it actually allows you to achieve a sort of ecological steady state, right, with your environmental and environmental resources. And so this could be one reason why evolution might have picked regulatory mechanisms that actually promoted also the process of aging. Okay, and this is sort of one of my favorite quotes from Teschendorf. In our ancestral species, natural group selection could have favored genetic and epigenetic mechanisms that actually promote the aging process. Not to be so negative about the outlook of this talk, but now Mike will tell us some things about possible treatments.
So what are you trying to say there, Jim? We've gone into some of the mechanisms associated with epigenetic changes in aging. Now I just want to show some of the evidence that, well, how do you know these epigenetic changes as we age actually, is associated or causative of aging? So one of the biggest findings in most recent years is cellular reprogramming. And this is a process in which you can actually create stem cells. So Tim had mentioned about embryonic stem cells very early on, how those can be, go from an immature state to differentiate into many different cell types. I talked a little bit about adult stem cells, which everyone has, that helps to replenish those cells in your organs. Well, induced pluripotent stem cells was a fantastic finding by the Yamanaka group, because this is where you could take a mature differentiated cell that is an adult cell, which has all those epigenetic marks, and then reprogram it, actually change it genetically with different proteins to reset the epigenome, reset the epigenetics, actually change those biochemical marks to make it think that it's no longer an adult mature cell, but actually a very early embryonic-like cell. So this is illustrated here. And those factors, there are 4 of them called the Yamanaka factors. And then so this has allowed us to really study the epigenetic process and also maybe a source of stem cells. So, I'm a little frozen here. Uh-oh. Oops, there we go. And this goes back to a very famous illustration showed here, which goes to this model from investigator Dr. Waddington, in which what you're really doing is modeling embryonic development. So very early on, when you have in a developing embryo for embryonic stem cells, that cell is immature and pluripotent. And you can think of it as at the top of this diagram. Okay, and then as it differentiates during development, it has these little valleys that it's going to go and decide what kind of cell it can become, and then become a unipotent or mature cell, as illustrated here. In embryonic development, that's called differentiation, which we have mentioned. What it's also doing, you could think of it as changing what we call the epigenetic landscape. What are those epigenetic marks that makes an immature cell allow it to become a more specialized cell? And from this, you could think of this process of cellular reprogramming mimicking embryonic development. So now you're taking a mature cell, a somatic cell like a fibroblast, reprogramming it, resetting the epigenomics into what we call an induced pluripotent stem cell, and then having the ability to take that IPS cell and differentiate it back into the mature cell. So you're actually modulating the chemical processes on the DNA that makes a cell go from immature to mature. Now, My lab uses iPSCs to study genetic diseases, and I'll just show you some of the examples of this technique where you could take a mature skin cell from a skin biopsy, and the picture here on the left is magnified, so that's a little dark area right there is a few millimeters based on a skin biopsy piece of skin, and then the cells would grow onto the dish, and then they become fibroblasts, which is a type of differentiated mature cell. It's growing on the culture plate. So if you could look carefully, these kind of spindle-like shapes are each a cell. And then when you give it the Yamanaka factors, then you can convert this fibroblasts into iPSCs. And then after that, you can take those iPSC cells that mature and then directly spontaneously differentiate them into different mature cell types, as shown here. As an example, ectoderm, it's a type of embryonic tissue that is staining here. Again, these are cells grown on a slide and then stained chemically to identify what kind of cell is there. So these went from an immature state into a specialized cell. In the first column here, it's a marker for early neurons, or brain cells. The middle column shows a marker for endoderm tissue, which can give rise to very early cells such as liver. And then the third column in red here is a marker for mesoderm, which can give rise to early cells such as smooth muscle. My lab also specializes in cardiomyocytes. So these, again, is a magnified image of those cells. The bright pink circle is the nucleus. And then you can also differentiate iPS cells into what we call multipotent stem cells. So such as mesenchymal stem cells, and these can be then differentiated into different types of cells. And these, again, are magnified differentiated cells stained with different chemicals. In the first column, it identifies very immature bone cells or osteoblasts. In the middle column, identifies very early adipocytes or fat cells. And finally, the last column shows staining for cells or chondrocytes, which make up cartilage. So this technique is quite powerful because now you could take you could take a somatic mature cell, convert it, or reset the epigenome, and then have a source for mature cells. Now, as we go through this, I want to give you an example of some of the treatments that are coming down the pipeline, or at least in the research realm. And we have talked about studying aging in terms of using DNA methylation, like the methylation clock, and cell replication. You could also study aging in the dish. Now, another way is to study patients. Now, this is a slide showing a picture of a boy with a condition called Hutchinson-Gilford-Progeria syndrome. And this is an early aging disease. So it's a very rare disorder. It's a genetic disorder that happens spontaneously. That is, it happens for the first time within the family. And for these children, within the first 1 or 2 years of life, they appear normal. And you can see this little boy here at age 1. And then around age 2 years of age, they start having growth failure. That is, they don't grow normally despite their eating fine. And then they start having a characteristic facial feature, as this little boy is shown here, in which the faces appear small compared to the head. And then as the child gets older, you could see in the picture here how the facial features become more prominent. So it's considered a model for premature aging. And it's due to this gene called lamin AC, which makes a part of that nucleus and plays a role in epigenetics processes. So there's another slide showing multiple children with this disorder. It affects both males and females. And as listed here, some of the features that these children develop mimic aging, with hair loss, hearing loss, osteoporosis, or weakening of the bones, muscle wasting, and they suffer from early death. The average lifespan is about 14 and a half years old. And typically that results from chronic diseases such as atherosclerosis or hardening of the arteries or strokes. Now, this particular condition, because it's a model for early aging, has had lots of areas of research. And one of it was developing a mouse model by this group in which they changed the lamin AC gene in the mouse to mimic what you see in the patients. So in this slide over here is a normal mouse, what we call wild type, and you could see the size of the mouse over here, and then also the shininess of the hair color. But when they've introduced that genetic mutation in the lamin AC gene to give the mouse this progeria syndrome, then you could see this is a mouse below that's affected that is the same age, but you can see how small it is. And then when you look at the hair, you could see the hair loss and some other changes. And when you look at the survival of this mouse, it's associated, the mutation is associated with early death. So this is a survival curve showing the age. Now mice live about 1 to 2 years of age typically. So this is measured in days, and then the percent survival. So those wild type or normal mice, those mice without the genetic mutation, all survive way past 1 year of age, but those here as signified with the minus or having the genetic mutation in red here, this line shows how roughly after about 2 to 3 months, then the survival drops quite readily. So it mimics what we typically see in the patients, okay? Now, animal models are important because it allows us to study processes such as these. Now, I bring this up because several years ago, a group down in San Diego actually had a very dramatic experiment in which the question is, if you can take these Yamanaka factors and reprogram a mature cell, that is an age cell, with all these epigenetic changes, and convert it back to an immature cell, can you do that actually not just in the dish, but actually in vivo, that is within an animal system? So this group, what they did was take this progeria mouse, as shown here, and then set up a very complicated system in which the cells of the mouse were reprogrammed using those 4 factors. Now, it's important to note that if you just give these 4 factors to any animal model system, the risk is early death and cancer. So what they had to do was actually do something called partial reprogramming, cyclic. So the cells actually were activated to give the 4 factors at a limited time, and then it was turned off, okay? Because when you try to reset the landscape of these cells, you don't want, of course, the cell to actually give up its identity, set it all the way back. So they partially reprogrammed, and what they did was quite dramatic, because when they did this to the progeria mouse, shown in this slide over here, on the left, the one that was reprogrammed, really many of the features improved, that is, were rescued. These are mice of the same age. You could see again the difference of the hair color, and then again survival over here. This is a complicated slide because just the way they administered the reprogramming factors had to be considered. But the most important thing here, again, this is a survival curve showing the age in weeks below, and then percent survival. That in the blue line, those mice with the progeria mutation and subjected to partial reprogram had significant improvement in survival. So this was, a proof of concept that if you modify your genome within the cell in a living animal, that you could actually reset it and rejuvenate the cells and rescue the phenotypes associated with aging. Now, the other thing they did was, that's the progeria mouse model. What about the mice at age? Does that have any effect on them? So they took some older mice and then did a similar thing. Now, these are mice, normal mice do not have the lamina mutation, they're older. And then they looked at the response to injury. As I mentioned, you need tissue homeostasis. So as you get older, it's really hard to bounce back from illness, from injury, compared to when you're younger. So what they did, they took the older mice and then subjected it to very specific types of injury to mimic some of the chronic diseases that we see with aging. One is diabetes, so when they injured the pancreas and then subject mice to this reprogramming. Now, these are older mice, normal mice. These mice would respond better given the reprogramming. That is, this is a model in which they're essentially trying to mimic diabetes, chronic diabetes, which involves the pancreas. So when the mice were challenged with glucose, the partial reprogramming actually allowed them to bounce back. That is, the pancreas regenerated better than those mice that did not undergo the treatment. The same thing with the muscle. They took these older mice, they subjected them to muscle injury, and then those mice that were partially reprogrammed were able to respond. That is, their muscle regenerated better than those without the treatment. So again, this was one of the proof of concepts that perhaps using methods like this, partial reprogramming and resetting your epigenetics, perhaps this may be a possible treatment to address aging.
Great. And so just to piggyback on what Mike sort of mentioned, although a lot of those methods are sort of in development, there are sort of more practical things that you could do now to help slow the aging process. And so this is a schematic from the same paper that I mentioned before about epigenetic drift across different species. And just to give you a flavor of what was done is they had mice and monkeys that were given ad lib diets versus caloric restricted diets, okay, so restricted eating. And what you can see is their actual age versus the predicted age based on DNA methylation sort of doesn't have a clear impact or change in the animals fed sort of ad lib diets. But when you have this caloric restriction, you can see that at the level of the epigenome in this epigenetic clock, you can actually see a reduction in the predicted age in both these animal species. And so this idea, you could hit the next slide, I think, is really sort of, emerging as a common sort of theme where the idea is on the side of the epigenetic clock, there are these things that will sort of lead to accelerated aging and certain risk factors like smoking and drinking and other lifestyle choices versus having a healthy lifestyle where working out and exercising, good diet can actually lead to sort of a healthy aging. And so, I'll turn it to Mike.
So as Tim had mentioned, one thing we were pointing out many of the basic science epigenetics of research. However, of course, these are many ways, these are avenues, potential avenues, So this, we wanted to end this talk just as a reminder for all of us. Actually, the best advice is having a healthy lifestyle, because there's actually many research, like the one that Tim showed with the caloric restriction, that does make a difference in your epigenetics and lifespan extension. And there's a long list of things that are actually being tested right now. One of the first FDA trials just happened about 2 years ago, in which they had individuals. This was a cohort that they were following of individuals between 40 and 60. These individuals had a very loose diet. That is, they didn't really watch what they eat. They watched a lot of TV and did not exercise. Okay, so I probably could have been in that cohort. Anyway, so they had an FDA trial in which they randomized them and then kept track. Did the DNA methylation clock first to see what their epigenetic status was, and then they randomized them to have a treatment group which consisted of diet and exercise, okay? And then the other one which did not. And after about 3 months and 9 months, when they rechecked the DNA methylation status, it actually improved. Okay, so there is, some direct evidence, and there are many studies coming down the pipeline, moving away from some of the theoretical concepts that we talked about and the animal models to actually patients, because now there's a very good way to measure aging with the DNA methylation clock, and many different lifestyle changes that you could do right now to implement and see if there's a difference. Okay, so. Can we stop the hands of time? Well, as we had mentioned, it's very promising approaches. Over the last couple of decades, we've really learned a lot about aging. The molecular changes, the cellular changes, the epigenetics. Epigenetic changes is a very proven hallmark of aging. And then now we're getting into the stage where actually, not only we can measure, but maybe we could intervene. Whether it's something like partial reprogramming or environmental changes, these are very promising, such as additional breakthroughs, are on the horizon. So again, we just want to emphasize, and as a physician, I just again want to mention that lifestyle changes is the best, a very good approach, and as the stem cell techniques are being developed. And we just want to show this. We stop the clock, and we thank you.
So, all apologies, sir, but I don't like your prescription of I want a pill. I want a pharmaceutical. So what can we look forward to, assuming that we're not going to exercise and we want to eat our In-N-Out burgers? Are we doomed?
No, we're not doomed. Okay, and this is when Tim and I was discussing what to present. This is a very broad. It's a rich area of active research. Certainly aging is a big topic, not just with academic centers of pharmaceuticals, but one of the big things, as Tim presented, was the DNA methylation clock, which is an important finding because now we can actually measure, aging quantitatively. So right now there are actually many, I will not mention specifics or what drugs, but there are active trials for drug regimens monitoring people with the DNA methylation clock to see if it makes a difference. Okay, so those are on the horizon, and we'll just have to wait. Okay.
So I think it's related to what you guys were just talking about, and I'm sorry if I missed it earlier. So I think you made it pretty clear that the methylation decreases with aging. Is low methylation associated with diseases or causing diseases, something like that?
Yeah, that's a great question. And so what we showed previously is there is a global hypomethylation that happens, but really it's a little more complicated what happens, because in the genome you do have regions that are highly methylated and regions that are lowly methylated. Oftentimes promoter gene regions can have no methylation present. And actually what you see during the aging process is that the hypermethylated regions lose methylation, but the hypomethylated regions actually start to gain methylation. And so in that sense, you can actually have gene dysregulation within the promoter regions as methylation is being hypermethylated there and usually associated with the silencing of gene expression. But then I think it's also interesting in other regulatory regions that are highly methylated, you do see losses in methylation, and I think those could have an impact on sort of misregulation of genes as well. But it can be kind of context specific or gene specific, but you do see both scenarios happening, but largely the genome is highly methylated, and so globally you see a loss in methylation. Yeah.
So I want to follow up on that. So like chair's prerogative kind of thing. In layperson's language, right? So you're losing methylation with aging. What is that doing for the cell, right? So is it that the cell is just making too many genes now and so they're not working together? Is it inefficient from an energetics point of view because there are changes in energy metabolism that happen with ages? Like, so why does it matter? Is it just that you have a finely tuned orchestra and now they're not all playing together?
You know, I think that last point can be a very big thing that we do observe. So, for example, there have been studies that looked at DNA methylation in muscle stem cells as we age. You have sort of an increased variation in the expression of genes in those stem cells with the also changes in methylation that you observe. And so it seems like these cells are unable to regulate their genome and ultimately the expression of genes throughout stem cell populations as well as they were when they were younger. And that could cause changes in the sort of differentiation capacity or efficiency and ultimately stem cell exhaustion that we sort of mentioned earlier.
So, and then, do I have a question from the audience? Cool. Oh, good, you've got one more.
My question is, you were saying that, if you have a good diet and exercise and all those little words that you had, so are you specific with, do you measure like, well, if this person is a vegetarian or meat, are you being more specific besides just all those words that you had up?
That's a very good question. And no, a complicated question, because aging is a complicated process, as we know. People age differently. The diets are different. The cells and tissues actually age differently. So until we get that magic pill, or we have specific ways to address aging, I guess always the general recommendation, and not just for aging, but for healthy lifestyle, is diet and exercise, stress reduction, sleep, because those are things that as you perturb your system, that's what's think is going to be changing whatever it is, whether it's associated with disease or aging. Okay, so but some of those questions are very, very good, and I think we're moving towards. Again, you hear the word of personalized medicine, because you could see the research is very broadly done from a conceptual basis, hypotheses, animal models, and then cohorts. Like the cohort I said with the first FDA approved, which is 2 years ago. It's a big thing, but that was only a very small study and very limited, okay? So until we get enough resources and focus on aging and how that really matters individually, I think that's when we're going to be able to address it, something like that. Okay.
You're not studying anything specific like medicinal mushrooms, not the
Yeah.
genetic ones. There's been a lot of talk about specific mushrooms.
Yeah.
Mike, can you repeat that question just so we know that people online are
so
able to hear?
there's also other interventions like medicinal medicines, other kind of interventional or integrative approaches. Those are all very good questions, but I'd have to go back to see what's in the literature. What we wanted to focus on was epigenetics, and at this stage, these are very broad studies in which they're looking at some of the low-hanging fruit, the obvious things that are often recommended just for general health, like decreased screen time and fruits and vegetables, caloric intake. But eventually, I think we're going to have to have a broader picture and see what kind of environmental stressors or interventions, including things like that. Okay.
But to your guys' point, having this methylation clock gives a tool where you can ask those questions, right?
Absolutely.
Whereas even a few years ago, you didn't have a tool to be able to figure that out.
Yeah, but I'd have to see in the literature to see what specifically, because we do have the tools in terms of the cell models, the measurements, and the hypothesis that you could imagine how to address that. But then actually taking that, even from an animal model to a person, is going to take a lot of effort. Okay.
So we have a kind of a natural follow-up in one of the Q&A questions that has come in online, and you just touched on it briefly, so I want you to expand. And that's whether different cells and tissues are differentially susceptible, right? Do they age differently? And then the follow-up question, which comes in the line after that, is whether that might be related to changes in the incidence of cancer, susceptibility to cancer of different cells and tissues with aging.
Yeah, just speaking upon the question of, do different tissues age at different rates? And actually, that's where a lot of the work with epigenetic clock development has gone, actually, with identifying sort of clocks for each specific tissue. And so you might find actually different epigenetic clocks in the blood versus the pancreas or the kidney. Actually, I have some collaboration with Marcelo Wood at UCI looking at hippocampus aging, and our hope is to find a DNA methylation clock specific to that tissue so we can make predictions about cognitive decline actually throughout the aging process. The second question.
Susceptibility to cancer, right?
Yeah, I mean, I think, I think that's a great hypothesis. There probably is certainly an association to susceptibility, tissue-specific susceptibility to certain cancers that are associated with those sort of rates of decline. In fact, a lot of the epigenetic landscape that you see during the aging process mimics somewhat that of the cancer epigenome as well. And so there is some link there. Yeah.
So there's, I think, another interesting question that came in online from Eric Swanson, I think, is about kind of stacking up and comparing the relative importance of methylation status and this demethylation or hypomethylation process that happens with aging to something people have probably read about in the past in Newsweek and Time Magazine, which is telomere shortening, right? And you touched on it, both of you, in your talks, but could you just briefly explain what telomere shortening is in the context of your answer also?
Yeah, so telomere shortening essentially is the result of what's known as the in-replication problem. So as DNA replicates, half of the genome has to be replicated in what's called Okazaki fragments. And that leads to a problem at the end of your genome where there's not enough DNA left to make a new strand of DNA. And so the cell, to sort of create a blunt end of DNA, will chop some of that end of the chromosome off. Stem cells express telomerase, which are enzymes that can build back those strands. But as we age, the levels of telomerase actually decline, and our ability, and even in somatic cells, they really start to sort of shorten, leading to genomic instability. These ends of the chromosome protect the genome from these chromosome instabilities, essentially. I think when it comes to making predictions on age, telomere shortening versus DNA methylation, I think that DNA methylation has been shown to be more accurate in those predictions, and so that's why there is a lot of excitement actually about this as a metric versus telomere shortening, but there are certainly companies that will give you some prediction based on telomere length as well.
So, an interesting question that also is posed online. I'm going to ask it in two different ways, and then there's a great sort of ethics question maybe to wrap up with at the end here. I'll give you guys one more chance before we close up to ask anyone who's present in the room. So that is, and I'm going to give you two sides of the coin. For current cell therapeutics that we're looking at, cell transplantation, for example, is it possible that one of the mechanisms, I'm paraphrasing a little bit here, but one of the mechanisms of those cell therapeutics has to do with actually changing the cells in the body in terms of their methylation status, right? So that's part one. And part two, which I'm adding on to the questions that came in online, is do we need to worry about, for cell therapeutics, monitoring what their methylation status is? Might that be related to the efficacy of a population or how we have to maintain it in a dish, something that we need to think about if we're going to be doing transplants for spinal cord injury, for Alzheimer's, for Parkinson's, for diabetes? Should we be monitoring that piece? Is it important, probably, in your view, towards function?
Yeah, those are great questions. The first one, remind me of the first one again, just briefly. It was.
At least I'm not Peter. I didn't ask you like 5 questions all in one sentence. So
I had it, but then I lost.
the first question is, could one mechanism of what a cell therapeutic does be to reprogram methylation status in its target tissue, like where you're transplanting?
No, absolutely. And I think that speaks quite a bit to what Mike presented.
Yeah, it was interesting with the research from the progeria mouse and the partial reprogramming. Now, one of the points is they're actually rejuvenating all the cells. So it's set up genetically so that every single cell of that mouse is actually having the 4 factors. So looking at the data there, you can't really say, well, was it actually stem cells that are being targeted and being rejuvenated, or was it the other cells, okay? What was very interesting was just the same group just very recently had a paper in which they looked at those muscles and then actually targeted specifically the muscle cells. And it was interesting that what happened was the muscle cells have very specific stem cells called satellite cells. And in those mice that had rejuvenated muscle due to the reprogramming, it was those stem cells were much more active. But it wasn't because of the reprogramming. It was actually from the surrounding cells that was actually contributing and activating the endogenous muscle stem cells. So given that, you could imagine that, as I mentioned with some of the aging process with adult stem cells, all these things can happen to change that. We also have to remember this is not an isolated system where you're just trying to activate the stem cells or replace the stem cells. You really have to pay attention to the local environment and also the entire body system. And that's what we really don't understand. We can spend a lot of time looking at certainly one of the major players, in our focus, which is the stem cell. But what about the environment that it's in? Those cells that actually have to maintain the stem cells, or in this case, activate it. And then also those circulating factors in the entire body, okay? Which there is other, and we didn't have any more time, but there's other evidence of circulating factors. So it's a very complicated system, and certainly if you're gonna transplant, the changes could actually be happening to the surrounding cells.
So I just want to remind everybody of kind of the coolness of all this when it comes to stem cells, right? Which is kind of embedded in what you guys have been talking to us all about this evening, but I think is easy to get lost on the audience. And that is every tissue in your body has a population of stem cells right now, right? And so on one hand, we think about ways to use stem cells, to transplant them, to improve function by making more of whatever is there that's become dysfunctional or missing. But embedded in this is the idea that we could wake up those stem cell populations that are in an aging tissue or in a diseased tissue and use that as a means to repair, right? Never transplanting anything at all. And how cool would that be down the road? And some of this research that we've been talking about this evening is going to be really important towards that process. So we've talked before in these meetings about the idea that you've got a really large base or pyramid that we're stacked upon, that's all the basic science research that leads up towards the translational path. And this is a part of what you've been hearing tonight. So on that note of coolness, I want to ask one last question that has come in from the audience. And that's from Rick Robertson, who's an emeritus, maybe it's from Sid, who's an emeritus faculty here, very active participant in the Stem Cell Center and engaged in the School of Medicine for many years. And he asks, from Jeff Goldblum's character, right, in Jurassic Park, because you can do something doesn't mean that you should. And so should we be thinking about reversing aging? Or is there an ethical implication to that that's on the con side of the game? I'll let them struggle for a minute, and then Lee, since you're here, you should probably chime in.
I can start. I mean, I think, it's a good question. I think it's society's to be decided in some sense. I think from my perspective, I'm very interested in preventative tissue engineering. I got into science really to build new organs when organs go bad, things like dialysis and kidney failure. It's not so robust, in terms of the long-term survivability of those patients. And so, I think about sort of the age-related disease aspect of it. And If it's natural, do we treat it? Well, I think we, all want to sort of extend that health span. And for me, it's really about health span more than lifespan. And so that's kind of where I'm at with the debate.
That's a tough question, because that's why in the beginning I wanted to bring up the question, is it aging a disease? Because if you call it a disease, then we should treat it, okay? But this, of course, one of the reasons I like to participate in these community talks is because to hear what the community thinks about this, okay? Because these are real active issues. You could see the research going, and there is a big push, certainly to address those chronic diseases which are associated with aging, okay? But ethically, there are some very good points here, okay? So it's very hard for me to answer that. As a scientist, I would agree with Tim, because I'm looking, and as a doctor, to preserve life, be healthy, and then push the lifespan so we can decrease the incidence of those chronic diseases, okay? But as a society, the question is, what about the bigger issues, okay? That's not for me to answer. I think this is a discussion between individuals, the community, scientists, because this is very important on where we direct the research, okay? It's very easy to see the expected life expectancy and say, hey, let's push it, okay? The other thing I should mention is there a maximum lifespan? That is an open question too. We could have these efforts and try to extend the lifespan, but there might be a limit to that, okay? So I'm not really sure. Does anyone else have any thoughts on that? Is aging a problem that we really need to direct our efforts to, extend life? Or, I don't know.
Lee Turner, I'm new faculty building a bioethics center here at UCI, and I enjoyed your talk, really appreciated it, thank you. Just to kind of pick up on the question that was posed, just because you can do something, should you do something? I'm guessing a lot of us would kind of quickly turn to the answer being no, that just because you have the technical capacity to do something doesn't automatically mean, that you should do it. That said, turning to the focus of your presentation, when it comes to the question of, well, if we could develop therapeutic interventions for children with progeria, for example, our response to that might be different than other aspects of human life. So I think a kind of a case-based, very sort of specific, focused way of looking at this might be a helpful way to go. Just to kind of throw in my own 2 cents here, I found myself thinking at the end of your presentation where you kind of moved to the language of lifestyle choices. I found that illuminating and helpful, but I also found myself wondering, do you think it might be helpful to complement that with a kind of, public health perspective or population health perspective? Because, part of your presentation included discussion of environment, and there's the environment for cells, but there's also our environment, the world that we live in. And if, your goal as a society is to try and have healthy aging for your population, for individuals, there are a lot of policy things that you can do, for your society. And if you have strictly a biomedical, highly technical orientation, you could end up with a scenario where you're kind of pouring more and more resources for less and less in terms of your gains, kind of like tiny little increments at the end of life where you're not really getting very much for your extraordinary allocation of resources. Whereas if you focus more on, making sure that there was healthy food available for most of your society, or there's, maternity leave, paternity leave, quality education available to everyone regardless of income. Pardon me? Clean air, you're like very concerned. Those might be sort of like well-established policy measures and public health measures that they don't stave off aging, they don't stave off death, but they might, for a population overall improve kind of, healthy aging over time. So it might be that there's sort of different disciplinary lenses and perspectives. The concern I have with lifestyle choices is it can be a bit too individualistic and make it seem as though it's kind of like it's all about your choices, like do you have the potato chips or the apple? But there are a lot of things that we can do to kind of like make that apple a possibility and make the potato chips a bit less of a possibility. And so, maybe that's a complement to your concluding remarks.
Any other questions or comments, discussion from the audience? Because following on from Lee's comments, I would just throw out the idea that, of course, the environment that we live in is going to influence what our health index is, right? Or what was the phrase that you used, Tim?
Epigenetic clock.
Yeah, epigenetic, well,
Biological age.
health span, that's where I was going, which is much cooler than the other two words that you just said. No, health span, right? And we know that. We know that pesticides and lead and toxic things in the environment and, you know, where we are with climate change is going to influence probably what our health span is. It'll be an interesting question how that feeds back to our stem cells and the biology of those stem cells. Thank you guys all for your attention. I know that we went kind of long tonight, but I appreciate all the questions. Thanks again for coming. Thanks particularly to Dr. Downing and Dr. Zaragoza for being here tonight, and have a good evening. Round of applause. I can't do that well.